From Wildlife and Fisheries, Dr. Roel Lopez. The title is provocative, right? I think he'll tell us about deer in love. I know he's interested in cats, squirrels, anything furry. Dr. Roel Lopez is an expert on. The title of the talk tonight, Chemistry of Love, Controlling White-tailed Deer Reproduction. It's a Texas topic, and Dr. Lopez is a Texas man. He was born in McAllen, Texas, as were 7 other people, and took his bachelor's degree at Stephen F. Austin in wildlife, and then did his master's and PhD at Texas A&M in wildlife and fisheries. He is an internationally known expert on reproduction, and it turns out his specialty is the key deer of the Florida. But his own research now takes him to NASA, and he'll tell you other topics as well. Thanks for joining us. We're happy to have you. Welcome.
All right. Thank you. Howdy. Howdy. I thought, Eric, I was at Texas A&M. Howdy. Howdy. Thank you. Okay, as Eric said, my name is Roel Lopez. I'm an associate professor in the Department of Wildlife and Fisheries. And this evening, I'd like to talk about an issue dealing with urbanization and overabundant wildlife, namely white-tailed deer. And there's going to be a tie-in, seeing that this is a first-year program for chemistry students, in trying to illustrate the importance of chemistry in whatever discipline, including wildlife conservation. So for my presentation, I want to start out by giving you a little bit of background on this idea of urban development, urbanization, and how that affects wildlife populations. I then want to use Johnson Space Center, NASA, in Houston, Texas, as a case study in trying to control overabundant deer or high deer densities. And in doing so, look at different methods available, lethal and non-lethal approaches, with focus being on birth control. So we're going to talk about deer sex and its relationship to wildlife conservation and tying in chemistry. So if that doesn't excite you, I don't know what will. Okay. Urban development. In the last 150 plus years, there have been more buildings, pavement, roads, etc. And that results in changes to wildlife populations. You know, all these subdivisions and so forth really fragment native habitats. And as a result, we have these patches of native habitats, these island refugia, if you will, that are beneficial to some species, maybe not to others. And so, in looking at urbanization, urban development and effects on wildlife populations, there's a couple of general trends that I guess I'd like to first introduce. And I'm going to give you guys a little bit of background and then go specifically into this research project that looks at this novel vaccine developed to control urban white-tailed deer. In general, when urbanization occurs, we have a simplification of habitat types. We have an increase in biomass. You know, for those that shop in College Station, all the blackbirds that are in the shopping centers and so forth, that's an example of an increase in biomass of certain species, a decrease in biodiversity. So overall, bird diversity within urban areas decrease. And we see some other unique adaptations in terms of exotic species, feral cats, pigeons, for example. But the main point here is urbanization, urban development affects wildlife populations in different ways. And I want to go through 3 examples of research projects that we've been involved with. The first is the Aggie Squirrel Project. Many of you have seen the terrorist-looking squirrels with the little collars running around campus. They're actually radio collars. And I had a student, a PhD student, that compared, for example, demographics. And just to kind of give you a little bit of background, I'm basically a demographer, a wildlife demographer, looking at survival, reproduction of wildlife populations in different types of environments. And urban development is one of these areas that I focus my research on. So going back to this squirrel project, the idea was really to compare squirrel demographics between an urban site, i.e. campus, and a rural site, the Navasota River bottom. And what we found was that survival is much higher in urban sites. Rural sites, it's much lower. This is actually a hawk that killed a squirrel on campus. So a lot of students got to see that. There's differences in mortality factors. For example, in the number one cause of mortality for urban squirrels, Aggie squirrels, is roadkill. Okay, the number one cause in rural areas is mammalian predators. And looking at reproduction for squirrels, urban squirrels have a higher frequency than natural populations. Take-home message is survival reproduction is higher for species like fox squirrels. Feral cats, we see the same thing. Had a student that did some work with feral cats, feral, semi-feral, and owned cats, pets, in Caldwell, Texas. And she found that survival increased as a cat became more urban. And looking at reproduction, same thing. Average litter size greater than 12 weeks increases. So the take-home message again is, as you become more urban, survival reproduction increases. And again, this is another case example. Now let's focus on our study of urban deer. Looking at survival for urban deer, for both sexes, in general, we see an increase in survival across, or for both males and females. And looking at reproduction, urban deer have higher reproduction, fawn-to-doe ratios than do, again, wild deer. And just to throw in another little statistic, this was actually some work done with key deer where we looked at changes in population demographics for key deer, which are an endangered white-tailed deer, over 30 years. And average weights for key deer have actually increased for both sexes. So here's the point. Urbanization benefits some wildlife populations. Survival is higher, reproduction is higher, and in some cases, you get fatter. We don't know of any other species like that, do we? That was a joke. Okay, I guess not. So the phenomena is what we call overabundant urban deer. And it's things that you normally don't think of. You know, so, I was meeting with a colleague earlier, and he was saying, well, so there's deer in subdivisions or in town, and you bet, eating garbage, getting hit by cars, things of that sort. And some of the impacts due to urban deer are damage to landscaping, highway mortality, threats of disease transmission, and of course, trying to balance the cultural conflict, because some people like deer, some people don't. So, urbanization. We've had changes in urban development for the last 100 years or so in this country. When we look at white-tailed deer populations, in the early 1900s, less than 500,000. We currently have about 16 to 18 million white-tailed deer in this country. At the same time, native habitats have changed to urban areas. Okay, so urban development has increased. There's been an increase in white-tailed deer populations, and collectively this becomes problematic. Again, deer, like other species, are very plastic, and so they're able to sort of adapt to these urban environments. Okay? Highway mortality is probably the biggest issue dealing with urban white-tailed deer. For example, there's over 200 human fatalities each and every year, and they cause about $1 billion in property damage. And how many people have ever hit a deer in a car? Raise your hand. Look around, quite a few people. So it's a big issue, highway mortality. Now, many of us may say, well, that's somebody else's problem. That's a northeastern problem. This is a Texas problem, okay? And unfortunately, we can't let Mother Nature take care of itself. We need to do something, okay? And again, let me just do another poll. San Antonio, raise your hand. Hollywood Park. Who has heard of Hollywood Park? Urban deer problems. Austin, raise your hand. Lakeway. Who's heard of Lakeway? A couple of problems. Kingwood in Houston, and so forth. So, I mean, there's many subdivisions that are having these kind of issues. And so, again, it's up to us to try to address these issues to try to minimize risk to people. Okay, so that's kind of the background. That's how we got here. Okay, so everyone kind of follow that. Now let's switch gears and let's look at NASA, Johnson Space Center. LBJ was established in 1962 to develop and test spacecraft, and it serves as mission control for the space program. They've also, like other places, have had urban deer problems, an increase in deer vehicle collisions, human-deer interactions, and collectively this is a safety risk. It's an issue that they want to deal with. It's about 600 hectares, about 1,500 acres or so, located on the east side of Houston in Harris County, and it's surrounded by a chain link fence. So it's a closed population. Okay, we've got 150 deer inside this island of habitat. That picture I showed you before, this is Johnson Space Center. You build a base and they will come. So we've got development and water on one side, and so we've got this population of deer that's in essence trapped. And I've mentioned that some species, like urban white-tailed deer, do well in terms of survival, reproduction. Well, guess what? We've got an increasing deer population. In 2003, Johnson Space Center wanted to do something about it. And hopefully I've made the case that this is an important thing for us to try to do. And so they wanted to look at feasible options, and their objective was to try to just maintain the deer population. They didn't want to get rid of all the deer. They actually liked the deer. They just didn't want 300 or 400 of them. They wanted about 100 or so deer. From a deer management perspective, there's a couple of options that we have. We can look at traditional methods, public hunts, sharpshooting, maybe even trapping and euthanasia. These are lethal methods, obviously. And in an urban setting, many times doing or implementing those kind of practices don't necessarily fly. They're not accepted by the public very well. And obviously doing nothing is not an option as well. And so really we're only left with two options: looking at birth control for white-tailed deer, or maybe trapping and translocating. So trapping the deer and moving them to some other place that maybe wants them. And so let's talk about those two in terms of options for Johnson Space Center. And again, these two were selected because they're non-lethal, humane, and they didn't want to deal with the public. Most subdivisions, urban areas are very aware of public attitudes and thoughts toward wildlife management processes or techniques. And so they want to kind of keep that in mind. So starting with reproductive control. Controlling survival means some sort of lethal method. So we have to focus on the reproductive side of population control. There's a couple of things we could do. Surgical sterilization would work for an individual deer, but we're managing a population. We're not managing one deer, we're managing 2, 3, 4, 500, 1,000 deer, depending on the subdivision. And so that's really the primary limitation. We can look at some sort of steroid regulation type treatment that unfortunately requires kind of an annual booster or every couple of months some sort of booster, and also is really not practical for a free-ranging wildlife population. You've got to treat them too often, and knowing what you've treated and what you haven't limits you. So another option in terms of reproductive control is looking at contraceptive vaccines. And really, let me give you a little bit of background. Immunocontraceptives produce antibodies against protein or peptide, and they disrupt the reproductive activity, is the way they work. They're kind of unique in the sense that, and I'll talk more about this, they're actually being treated with healthy, normal deer. You know, typically when you think of vaccines, you're trying to target a foreign object, right? Here we're trying to basically attack itself, and that sort of makes it difficult. But the current form is something known as PZP, which has been used for white-tailed deer and is very successful. The drawback, like the endocrine regulation methodologies, it requires annual boosters. Okay, so again, this is still limiting in free-ranging white-tailed deer. And I mentioned some of the challenges in some of these contraceptives. Having to administer to normal individuals, and this again is sort of a unique challenge to investigators. So, how do we solve this? For those that are deer hunters, by the way, this is an urban deer at Johnson Space Center. So, kind of a big issue, no pun intended, in trying to control deer numbers. Chemistry, of course, is how we can do that. And of course, you don't have to be the mad scientist. Really, some technology that was developed 40 plus years ago includes liposomes. And liposomes basically, just for simplistic terms, can take a drug, let's say like PZP, and encapsulate that drug. And so instead of having just a one-time shot and having to administer an annual booster, using liposomes, which are microscopic fluid-filled pouches with walls made of phospholipids, you can in essence extend the life of that vaccine. Okay, so it's a relatively new technology that was applied in the case of white-tailed deer to trying to control mammalian reproduction. Originally it was developed for seals, and they found that it's worked on other species like elephants, wild horses, because as I mentioned, there's several species that have issues dealing with overabundance, including white-tailed deer. And the way Spayvac works, it basically, you treat the females, and PZP antigens are released gradually via the liposome, and antibodies are produced which then adhere to the reproductive sites for the ovary. And so in essence, it blocks fertilization. That's the way it works in a real simplistic way. Okay? The unique thing is Spayvac, and again, just to emphasize, this is where the chemistry, just basic chemistry, is really critical in, let's say, addressing a wildlife conservation problem. We went from an annual booster to a single inoculation that lasts up to 10 years. Okay? Our study is the first study evaluating Spayvac on a free-ranging wild white-tailed deer population in the United States. And so it's been found to work up to 10 years in seals. How long will it work for white-tailed deer? So far, 4 years. Next year, maybe I can come back and tell you it's 5 years. The point is it's worked pretty well, and the fact that it's only required one shot is really revolutionary in terms of overabundant population control. We have found that it's been 100% effective. Okay, and again, it really the take-home message is it offers wildlife biologists, land managers, an alternative to controlling urban deer populations. So this vaccine has tremendous promise, and it's something that we want to continue to explore. Okay, so the vaccine's produced by a company in Canada, and the gist of the project, and I'm going to now switch gears and talk about the results and sort of trying to tie all of this together and looking at the demographics of Johnson Space Center. Okay, so the objectives of the original study were to look at demographics, i.e. survival, reproduction, pre and post treatment. And again, seeing that this is a research project, we're trying to evaluate, again, the efficacy of the drug in controlling deer numbers or maintaining deer numbers in the case of Johnson Space Center. And in order to do that, we also wanted to develop a population computer simulation model to basically integrate this information into the model and evaluate different management strategies or scenarios. Okay? This type of program is very expensive, and I'm going to talk more about that in a little bit. And so we want to treat just the right number of deer so we don't have a deer population go extinct. That's not what NASA wanted. They want to maintain the current deer population. And so using that deer model, we can evaluate these scenarios a priori before we actually implement it. Make sense? Yes? Yes, yes. Okay, let's go. All right, so what we did is we trapped deer using drive and drop nets. Of the 60 deer we caught the first 2 years, we treated 48 with Spayvac, 11 placebos, and we radio collared and radio tracked those deer for 3 years now. And those numbers have actually increased now. We then used or estimated survival using radio telemetry data. We also estimated deer density using weekly survival or surveys, deer counts. We looked at reproduction via the surveys as well as walk-ins using the telemetry. So again, we're trying to gather information. How well is this vaccine working? Using the information, we then developed a sex and stage structured model to, in essence, manage or simulate these different scenarios. And this is the model. This is kind of a conceptual model. We've got males, fawn, yearlings, adults, females, fawn, yearlings, and adults. And part of the females, there's also some treated adult females, okay? So once they're treated, obviously there's no reproduction, and so they kind of go into a different box, okay? So that's sort of the model. 11 different or 8 scenarios. No management. What if we did nothing, NASA? This is what's going to happen. And then we evaluated different rates of treatment using the contraceptive. Single treatments of 50 and 75%, as well as annual treatments of 10, 25, and 50% for 3 years. Okay? So that's one treatment. The other treatment was looking at trap and removal. Okay, so instead of treating and leaving the animals on site, what if we just trapped them and moved them to some other ranch? Okay? Annual treatments, 25 and 50%, again applied for 3 years. So similar to the vaccine, but it's just a different treatment. The deer that stay there obviously can still continue to reproduce. Now for each scenario we ran 1,000 simulations. We incorporated stochasticity, and that's something that you don't necessarily need to know. But it's basically bringing in all of the different variability, if you will, in a system. By running 1,000 simulations, we can then look at averages over 5 or 10 years to see what would happen on average, okay? That's what all that jargon means. And in evaluating or comparing the scenarios, we wanted to look at long-term trends, and we wanted to look at ending populations. In other words, what's the average population size going to look like at 5 and 10 years, for example? Because these scenarios we ran for 10 years. They want to look at a 10-year timeline. So just to recap, the model is basically saying this is the deer population. If we were to do these things and grow the population for 10 years, this is what's going to be the outcome under a given scenario. And again, it allows us to then compare and evaluate at what rate we should be treating. Okay, so that's kind of the general idea. Survival, female survival higher than males. Females are much smarter than males, if you haven't figured that out, and so they tend to survive. That was a joke. Okay, thank you. Appreciate that. Reproduction, I've mentioned efficacy of the drug. It has been 100%. Of the control group, 73% of the control deer, the ones that weren't treated, had fawns. And looking at the fawn to doe ratios over time, we started pre-data. Actually, we started the project in 2001. And so the project or treatment started in 2003. And so fawn to does, this is the number of fawns per adult doe, have decreased dramatically, and we've maintained a pretty low fawn to doe ratio. So again, that's pretty high. This is sort of the average, about 1 to 1.5 fawns per doe. And so the half fawns kind of get together, they make a fawn, and off they go. Okay, you are listening, good. Density, again, I mentioned we estimated about 150 deer on Johnson Space Center, so a moderate deer density. And looking at population trends, we started treatment of Spayvac there, and population trends have declined and have stabilized. So again, what we're doing here is We've got a drug that says it does this. We're testing that. We have got a model that predicts these things, and we're sort of validating what the model says should happen. Okay, so the result is basically validating our model responses. These were the predictions. And looking at ending population sizes at 5 years, if you don't do anything, NASA, you're going to go from about 150 deer to 250 deer in 5 years. 10%, 25%, and I need to look at the numbers here, and 50% annual treatments for 3 years, single 50 and 75, and this is trap and remove, 25 and 50. Okay, so the take-home message is basically you've got to be trapping or treating about 25 to 50% of the deer population annually, okay, in order to have or maintain the no growth in the population or trap an equivalent number. In terms of the single treatments, you just have to trap a lot more. In other words, you could stretch your workout over 3 years or do it in the first year. It's up to you, okay? And looking at 10 years, basically the same kind of thing. We're just seeing more variability because it's harder to predict what's going to happen, year 5, 7, 8, 9, etc. But it's the same general trend. Looking at trajectories, what the population is going to do, it's going to increase up to carrying capacity about 275, 250. And if we do single treatments of up to or greater than 25% or annual treatments greater than 25%, we're going to maintain the deer population. Okay? So that's what the model predicted. And what I showed you, we've actually applied that rate based on the model results, and we're seeing that in the overall deer numbers. So the recommendation that we made at NASA was applying single treatments greater than 25%, and we focused on that because it's easier to catch, 25% of the deer population in a given year. Trapping deer over time gets harder and harder. You've caught the dumb ones, and year 3 it's harder to catch that deer. Okay, so from a cost perspective, this is all really very relative, and I hope you can kind of see that because again, we want to try to minimize our efforts, but still maintain the objective of our study. And again, the objective was to maintain deer numbers. And so we recommended 25% to NASA for their deer program. Okay? So like many urban communities, Johnson Space Center is interested in controlling white-tailed deer numbers. And the introduction or the development of Spayvac, this vaccine, that again required somebody that knew a little bit of chemistry. They weren't necessarily a chemist per se, but in our case, knowing and appreciating chemistry is really critical, and hopefully I have shown the relevancy of that. Okay? You may not necessarily be the best chemist, but understanding the basics in chemistry are relative in being able to identify possible solutions or talking to the right people. Okay? And so that's kind of one of the points I wanted to try to make. So development of Spayvac, really using liposomes, really has made this a viable option. And this is new. We're talking about something that's just been developed or been around for about 3 or 4 years. Prior to that, we had no options. And if you don't believe me, read the papers. Look at what's happening in urban communities. For those that said they were from San Antonio or Austin, I think you know what I'm talking about. I mean, and it's very controversial in how to address overabundant white-tailed deer. So Spayvac offers us an option. It's not for every population. If you have a fairly large population, it's not going to necessarily be effective. Or if you've got any kind of movement, it has to be a closed population. And we could talk more about that in a little bit. So currently, that's what we're applying at NASA, and again, we're actively managing that deer population. Look at the costs, $400 per deer. You think a computer model is important? If you got to treat 100 deer versus 50 deer, that's a $20,000 difference. So it's very important in knowing how many animals you have and what rates you should be applying this vaccine. Okay? So, again, this is a fairly expensive project in managing a fairly small white-tailed deer population. How many would want to spend that kind of money in their subdivision? Raise your hand. A few people? Nobody? Okay. Well, I mentioned another option. Remember? I talked about translocation. And I'm going to talk about that. Translocation is another option. But some things have happened. Initially, Johnson Space Center wanted us to trap and move deer to other places. That was part of our original charge, if you will. And in 2003, we had a chronic wasting disease concerns in Texas. And CWD is a contagious disease that affects deer and elk, and it causes basically the brain to deteriorate. Okay? CWD is a really big issue. You know, it causes your brain to shrink, you lose your bodily functions, and you die. Some of you are probably thinking, man, I've got CWD. You might. Actually, you can't catch it. Humans, it's not contagious to humans. Deer, white-tailed deer management in Texas is a big business. It's a multi-million-dollar business. Okay, so having the threat of CWD coming in from New Mexico or Colorado, and for those that know a little bit about some of the issues with elk and mule deer, know what I'm talking about. The way you treat CWD is if you've got a hot spot, you go in and you shoot every animal within a 20-mile radius. That can have a pretty detrimental impact on a population or on a community, right? If white-tailed deer hunting or management might be a part of it. Okay, so CWD is contagious, and so Texas Parks and Wildlife doesn't want you to trap and move deer to somebody else's ranch. You see what I mean? That's why we couldn't translocate deer in this study. Okay, so finding release sites was a big problem that we needed to try to address. Now, I have an idea, and I'm going to kind of throw it out to you guys and see if you think this might be a possible solution. In 2004, there was a food shortage at the International Space Station. You guys remember that? And so what I'm thinking is maybe venison for astronauts. You know, we saw some of the earlier deals on they're wanting burritos and stuff like that. I thought, well, maybe venison for astronauts might be a thing that might be of interest. Again, these are just ideas. Why are you guys laughing? This could happen, right? So I put, you know, I'm an Aggie professor, and so I did some math, and I said, okay, to move 75 deer, it cost about $8.1 million. So that's about $108,000 per deer. Okay? Now all of a sudden Spivak looks pretty good, doesn't it? So you see, my point is, and of course that was not meant to be serious, in the translocation, we can't move deer. And really, for NASA, we're down to just one option, and that's contraceptives. And so Spivak has shown a lot of promise, and really from a cost perspective, it works fairly well. Okay, so chemistry really is really important in wildlife conservation, and I've shown you one example of how chemistry has, in essence, opened up many doors and has given us other options. And really, whether you're a major or not, having that kind of background can help you understand some of the issues in your discipline or other disciplines and apply to wildlife damage issues, wildlife disease issues, ecotoxicology, for example, for those that might be interested, or our natural resources. Those are all areas or disciplines that require a basis in wildlife or in chemistry, okay? And with that, I'll kind of conclude, and these are just a couple of websites in terms of positions and so forth that you know, offer or require some sort of chemistry background. Okay? I'm sure there'll be time for questions, and so how do you want to do this, Eric?
Well, let's thank Dr. Lopez.
Okay. Okay.
Just a few short words before we adjourn, and for students who are interested in meeting Dr. Lopez, please come down. We have for you, as a token of our gratitude,
Thank you.
your poster. In addition to seeing how chemistry can relate to a field as what I would have perceived to be as orthogonal as wildlife and fisheries, I think you get a sense for why Dr. Lopez has an international reputation in the field. His passion comes through, but clearly to be good at this topic, I mean, computer modeling, math, wildlife and fisheries,
Right.
I mean, it takes a diverse skill set to be successful, and a school like A&M will get you that skill set. And what I hope you recognize is that the excellence that is going to be demonstrated tonight on the basketball court in Austin against T-SIPs is actually matched in a number of the faculty that you'll have a chance to interact with over the course of your undergraduate experience. And so take advantage of the excellence in both arenas. Thanks again, Dr. Lopez.
Okay. And I might take questions. You know, for those that would like, and I know some of you may want to leave or what have you, I'm open to taking questions, so you can holler them out or what have you. How did I do on time? Was I short?
It was right around 40 minutes.
Okay.
And so that's,
Okay.
given everybody's commitments tonight, it was perfect.
Yes, sir. Like PZP and so forth? And moving them, if you could, that would be cheaper. But it's not an option. A question that was asked is, are other methods cheaper, for example, sharpshooting? And the answer is yes, but there's certain places, like NASA, you can't, there's too much risk in discharging a firearm. And that's typically true in urban areas. And so they discourage that. That wasn't even considered. And that's true with a lot of subdivisions. You know, for those that live in Hollywood Park, you don't want to discharge a high-powered rifle. Okay? So that's a good question. Yes, ma'am. The question is, are they available for private ranches? And the answer is, they would be, but CWD prohibits that. In order for you to move deer belong to you, the citizens of the
vaccine.
Oh, yes, it could be. It could be, or a form of it. So I could give you some information. You can Google Spayvac and find out about it. Okay, so it's a pretty well-known vaccine. Any other questions? Yes, sir.
These deer that you give contraceptives to, can you go ahead and eat the meat afterwards, or you know that?
That's a good question. The question is, can you eat the meat? Part of the problem is it's a new drug. It hasn't been approved by FDA. And so what they're actually doing is, when we trap and collar them, we've got to put a tag that says, experimental animal, do not eat. Okay? And it's not believed, it wouldn't cause any problems, but we just don't know. Okay? So until it's approved by FDA, it's going to limit its broad-scale use.
But wouldn't also those deer that are the beneficiaries, I guess you could say, of that Spayvac, they're not going to be the ones that are typically going to be eaten anyways because they're in urban areas?
Right. Well, and typically with urban deer and so forth, as you brought out, they're not, they're not actively hunted or what have you. These are, many people view them as pets and so forth, so it's just trying to maintain numbers. Yes?
What placebos?
The placebos were just to kind of go through the whole process. Trapping, handling, did that cause additional stress that may have aborted a fetus or what have you? And so using a placebo just acts as a true control. Because without a placebo, then someone, in terms of a review or what have you, would say, well, you didn't harass those deer. Because if you've ever seen deer trapping, it's folks that say there's no stress, there has to be stress, and there is.
Is this problem big enough, like, let's say for Texas, would they subsidize the cost of these vaccines for like municipalities?
Actually, what they're doing now, Spayvac has formed a nonprofit group, and they're trying to, in essence, do that via the federal government. And so those are some of the things they're trying to work out. The thing that, again, that's unique is it's so new. We haven't gotten there yet. Right now, it's an experimental drug. So it would be real hard to get for just public use. You'd have to follow or fall within certain guidelines. We were asked to control deer, for example, in San Antonio at one of the power plants there. They've got about 200 deer in a real small area, and we couldn't get approval because of certain conditions for that. So it's a pretty tough process until it gets FDA approval. Remember mad cow disease and how it shut the border off between Canada? This is a Canadian company, and it's using animal parts, part of a pig, in the vaccine, and so it becomes problematic. Again, they've got to get approval. Yes, ma'am.
Do the enzymes or in the contraceptives, are they allocated in condition?
I'm not real sure on how it works outside of just the basics, because it's a company, they've got it trademarked and so forth, and we know very little about the vaccine itself just because they're marketing the thing. It's a multi-million-dollar drug.
Well, please join me in thanking Dr. Lopez
Okay.
again.
Thanks, guys. Okay. All right, man.